Well, it is my enormous pleasure to welcome you all to Cold Spring Harbor, the home of transposable elements. And it's so good to have everyone back again here in person. Though I just want to say, of course, masks are strongly encouraged. Your chair is not giving a very good example, but I do have a mask. When you're speaking, you don't have to wear a mask. But it's wonderful to be here. There are very few announcements. There's only one that I'm particularly fond of, which is that there is no parking on Bungtown Road. I really enjoy saying that. You've no idea. That has been said at Cold Spring Harbor meetings for more than half a century. So welcome to Transposable meeting. Tonight we have our keynote speaker, one of our keynote speakers, followed by the first session. So it falls to me the great pleasure of introducing Kathy Burns, our first keynote speaker. She really needs no introduction, especially to this audience, but I'm going to try. So Kathy did her PhD and MD at Baylor, and then famously moved to Johns Hopkins, where she did a fellowship and her residency, and started her famous work on transposons, in which she's really been a pioneer in the role of line 1 elements in cancer. She rose through the ranks at Johns Hopkins, as you all know, to become a professor in clinical pathology, and among numerous awards, including the Burroughs Wellcome, which was her first junior faculty career award, I think. I only picked out one very recent. She's been a member of the American Society for Clinical Investigators for the last couple of years. Of course, she then moved on to Dana-Farber, where she's now the chair of oncological pathology and also a professor at Harvard Medical School. She's really pioneered the role of line 1 elements in human cancer and in human disease, using all sorts of genetic and molecular tools, including some of the first GWAS studies in transposons, and also famously, of course, the ORF1 monoclonal antibody, which turned out to be an enormously powerful marker for all sorts of different cancers. It's a great pleasure to welcome her here. She's now, of course, the chair of pathology at Dana-Farber, but when I was doing a little web search, I noticed that she's also on the Find a Doctor website. She's actually very much a practicing clinical hematologist, and it's so wonderful that we could get an appointment with you tonight. So thank you.
Okay, great. Thank you very much for the kind introduction. Maybe it's appropriate because I will be sharing some patient stories with you all this evening. I titled this talk, A Feeling for the Human. A few of you asked me about this, so just in case the reference requires any explanation, let me see if I can advance this. A feeling for the human is my adaptation of a title of a book about Barbara McClintock, a beautiful biography written by Evelyn Fox Keller called A Feeling for the Organism. And so tonight I'll be talking to you about the organism that we are studying in our research group, and I'll try to highlight some of the great strides, I think, certainly not just my lab, but others as well in the field have made towards an understanding of the impact of transposable element biology in humans. So here's a picture of the book and of McClintock and of Keller. The title of the book comes from a quote that was attributed to Barbara. I thought it was beautiful and that I might read it here to get started. What is it, our author asks, in an individual scientist's relation to nature that facilitates the kind of seeing that eventually leads to productive discourse? What enabled McClintock to see further and deeper into the mysteries of genetics than her colleagues? Her answer is simple. Over and over again, she tells us one must have the time to look, the patience to hear what the material has to say to you, the openness to let it come to you. Above all, one must have a feeling for the organism. One must understand how it grows, understand its parts, understand when something goes wrong with it. And I would say to you that those of us who study human disease, and really all of us, have a very intimate familiarity with humans, which can make them very special model organisms. So tonight I'll tell you the history of human transposable element biology in the story of 4 patient cases. They're all compilations of reports from the literature. Some you will recognize. Others are compositions that I've put together based on patients that I've seen. Our first patient, number 1, is a story of a young boy who presents with bleeding. A 7-month-old infant presents with an anterior forearm bleed. It's excruciatingly painful, it seems, in examining the infant. The pediatricians have immediate concern for acute compartment syndrome, a condition where the space in the closed osseofacial compartment is filled completely with blood. There's a very large hematoma that's evident under the skin. Already on arrival, there is a presumptive diagnosis of hemophilia made. Surgery is not such a great option for dealing with this. The team is concerned rather about hemostasis. They begin transfusions of fresh frozen plasma and cryoprecipitate. These have concentrated coagulation factors. It is the early 1980s. The treatment risks exposure to HIV. In the subsequent years, more than 4,000 of the 10,000 hemophiliacs in the United States will die of transfusion-related AIDS. Our patient is one of 240 who go on to be further studied by a group of geneticists and pediatricians at the Johns Hopkins Hospital, including Haig Kazazian and his colleagues. They recognize in doing southern blots for the factor VIII coagulation gene in these children, the occurrence of an abnormal band on southern blot representing the insertion of a line 1 sequence in exon 14 of the coagulation factor. They write in their paper, L1 elements were found in exon 14 of the factor VIII gene, actually in 2 of these 240 unrelated patients with hemophilia A. Both of these insertions, 3.8 and 2.3 kilobases respectively, contain 3' portions of the L1 sequence, including the poly A track, and create target site duplications of the factor VIII gene. In addition, their 3' trailer sequences following ORF2 are nearly identical to the consensus sequence of L1 cDNAs. Haig and colleagues write, the insertion of line 1 elements involving retrotransposition of DNA sequences through an RNA intermediate into a new and distinct location in the genome represents a mechanism for mutation to produce human disease, fundamentally different from those previously described. What they were referring to, of course, was the mobility of these elements that make up so much of our genome. Line 1 sequences make up nearly 20% of our DNA. Signs mobilized in trans by those elements, nearly another 20% of our DNA. Frequently dismissed as junk sequences, these highly repetitive, technically difficult sequences to study had been largely ignored by biomedical researchers, and indeed, even after that case report, were largely relegated to infrequent studies, infrequent examples of human disease being caused by a new integration of these elements. We have learned as a field through many people's observations that the L1HS subfamily of elements and the ALU-Y elements that it transposes continue to be active in humans, and that our evolutionary history is rich with their activity, that much of our genome is a testament to their activity. And work by Christine Beck and John Moran and others have really showed us that it's the young and active elements which maintain variant allele frequencies in human populations and continue to drive the activity of these elements. So here's what we understand about the life cycle of line 1. Our understanding is that most of the sequences that we inherit are not competent for retrotransposition, but on the order of 100 sequences that each of us inherits has intact open reading frames and may be transcribed. However, typically their transcription is kept restricted by methylation of the line promoter sequence. When that mechanism fails, you get a bicistronic so-called unit transcript, an RNA intermediate that's very difficult to directly detect. We'll come back to this problem a bit. Really one of the best ways to understand that line is being expressed as a mobile genetic element is by looking for the proteins that then get translated, particularly the ORF1 protein, which is most straightforward to detect, although you can also detect the genetic activity of the second protein, the enzymatic function of the retroelement. The reverse transcriptase and endonuclease encoded by the ORF2p protein is responsible for line integrations like the one reported in our patient. One of the first things our laboratory was able to then come in and do to expand our appreciation for this activity was in collaboration with my postdoc early faculty transition mentor, Jeff Buca. I developed strategies along with a fantastic graduate student, Lisa Wong, to comprehensively map line 1 integrations in the human genome. This can also be applied to ALU-type sequences. Now, of course, we do this by next-gen sequencing, increasingly by long-read sequencing. At the time, we were using tiling genomic microarrays to detect PCR amplicons off of the youngest subfamily of L1 insertions. That gave us these beautiful maps of line integrations in this particular individual. And you can see the blue lollipops sticking out on every side of the chromosomal ideograms. Actually, at the time, were line elements that we considered novel insertions, not part of the reference genome assembly. It quickly became clear many of these were segregating common variants occurring in human populations. So then the question really became, as our catalogs of these sequences grew, what was the functional impact of all of this activity in terms of the genetics of you and I and the typical individual, not the rare individual who had an inherited line integration crippling a gene? And so that brings us to patient number 2. Patient number 2, who I'll refer to as unlucky. Patient number 2 is a 9-year-old girl. She presents to us feeling tired, weak, feverish. She goes to her pediatrician. She's given some antibiotics. Symptoms are pretty nonspecific. She continues to worsen, getting dizzy, developing bruises in gym class, and ultimately a nosebleed that brings her family to the emergency room. A CBC, a complete blood count, raises concern among the clinical team for acute leukemia, and they perform a bone marrow biopsy. She is one of 3,000 patients who will present this year with the most common form of pediatric cancer, childhood leukemia. She undergoes multiple rounds of chemotherapy as these precursor B-cell blasts have taken over her peripheral blood and her bone marrow. Her hair falls out. She has a bone marrow transplant at the disease relapse. It's the 1990s. This is one of the best types of leukemias for anyone to get, and cure rates in the 90s are rising. Nevertheless, our patient relapses at age 11, dies of infections. Her devastated family and her 7th grade friends are beside themselves. One of the children in her science class asks the question that most of the teachers and guidance counselors suggest is a question that shouldn't be asked, and that is why her and not me? There are other people asking this question. Her sample is one of 317 that make their way to the St. Jude's Medical Center, where Mary Relling and her group of investigators conduct a genome-wide association study to identify common variants segregating in human populations that create risks for diseases like this girl's leukemia. They have more than 17,000 control samples in order to identify discrete loci where the inheritance of a risk haplotype creates a predisposition for leukemia. They discovered 2 single nucleotide polymorphisms in the ARID5B gene, which is a gene that encodes a transcription factor expressed in the B cells that we just saw, that differed between the ALL, or acute lymphoblastic leukemia group, and the non-ALL groups with very high degrees of significance. The haplotype also distinguished hyperdiploid ALL from other subtypes of the leukemia, and the team concluded that germline variants that we all are at risk for, and many of us inherit and pass on, affect susceptibility to and indeed specific characteristics of ALL. Years later, our group at Hopkins came along looking for transposable element integrations that may be causative for conditions like this. And we identified what we think from a genetic perspective is a very good culprit in an ALU integration, so a non-coding sign retrotransposed by line in the ARID5B gene. The ALU insertion is flanked by trait-associated SNPs identified by the group at St. Jude's in the intron of this transcription factor. If you walk along chromosomal positions into the gene, the ARID5B gene, there's no risk of disease given the haplotype that you inherit until you get to that ALU integration. And then the nearby SNPs and the ALU itself, which we imputed in the genotyping cohort from St. Jude's, really represent a pinnacle of disease risk as soon as you move past the ALU and into most of the coding sequence of the ARID5B transcription factor, you return to baseline risk. We don't know exactly what it is about this ALU integration that's affecting the expression and activity of ARID5B. To us, that remains still mysterious, but it seems a very good candidate for the cause of this risk to, again, develop this very common form of childhood leukemia. It wasn't the only example. We found really scores of ALU integrations co-occurring physically with genome-wide association study signals identified by GWAS groups investigating a whole variety of different human diseases. Now, in each case, we haven't demonstrated the high degree of linkage disequilibrium that exists between the trait-associated SNPs and the ALU variant or the line variant, but I think that our work really highlighted a need for the continued consideration of these types of sequences as common risk alleles for human disease. I'll show you a couple of additional examples that we reported in subsequent years where we have a better handle on the molecular mechanism. Here's a great example of that. An ALU integration event at the CD58 gene occurs very close to an exon in the gene and is tightly associated with risk for developing multiple sclerosis. It turns out that this ALU integration represents something that affects splicing of the neighboring exon, so that when you have an ALU insertion, you have a degree of exon skipping. You have a favored isoform of CD58, which encodes, we believe, a non-functional protein involving a frame shift right as this exon is deleted. We were able to demonstrate the effect on splicing in reporter assays, and the locus is also a splice eQTL. Here's another example, an inherited ALU invariant at the CLEC16A gene, which we identified lands at a region that looks like it has important regulatory potential. It also, I believe, is an eQTL for this locus. Patients who inherit the ALU integration have a disruption of this enhancer function and lowered expression of luciferase in reporter assays and lowered expression of CLEC16A in cell cultures where we edit the ALU integration. So to advance and comment further on our understanding of inherited variants at this point, I think it's been clear in the years that followed the initial report from Haig Kazazian's group that rarely, but recurrently, there's more than 100 of these types of case reports, mobile element integrations will generate loss of function alleles that can cause an overt sort of example of genetic disease. But commonly occurring insertion polymorphisms may also impact heritable disease risk more broadly. They likely do this through effects on area genes, and we have examples where they alter RNA splicing and gene expression patterns. So another point that I would like to draw out from our first couple of patients is this concept that comes up again and again, I think, in medicine, and that is that a rare disease, a rare patient, an overt example of something that goes wrong with our biology can serve as a means to experimentation, investigation that reveals a pathway that is fundamentally very important, knowledge that's much more generalizable and broad than the initial sort of patient who presents to you. Again and again, it's used as a reason to better understand rare disease, which I think is a great segue to our third patient I will call our mosaic. Patient number 3 is a 52-year-old Japanese gentleman. He works as an airline pilot. He has a son in high school. He's normally pretty reserved, pretty quiet. That wasn't true when his boy was younger. They would play endlessly in very rambunctious and fun-loving ways. One day, he is on a long flight, and he develops back pain. It becomes excruciating over the next several hours. He stops in an area emergency room, gets some painkiller, is a passenger on a flight home. X-rays reveal lytic lesions, biopsies, metastatic carcinoma, colon cancer. He is one of 150 cases evaluated at a Japanese hospital, identifying line 1 insertions that occur somatically, and in this individual affect the APC tumor suppressor gene. APC is a critical tumor suppressor gene in the colonic epithelium. It's an early loss in many cases of colon cancer. The investigators discover a line insertion, again, directly in the exon of the gene. It can be, they write, expected to dramatically alter the gene product inasmuch as it occurred in the exon in the middle of the gene. It will be of interest, they go on to say, to determine the frequency at which line 1 mediated inactivation of tumor suppressor genes occurs in somatic cells. Indeed, that was the first example, but now our literature is filled with a myriad of examples illustrating that somatic retrotransposition is quite commonplace in epithelial malignancies. Colon cancer is not the only type, but in many of the P53 compromised epithelial malignancies, which serve as the most really devastating forms of cancer in terms of the frequency of their occurrence and the associated morbidity and mortality. This first really came to greater light when Scott Devine's laboratory began comprehensive line insertion site mapping in tumor versus normal samples. We contributed to this literature looking at pancreatic ductal adenocarcinomas and high-grade serous ovarian cancers. This Circos plot just shows you locations of somatically acquired line integrations. Same thing with these red hash marks in a case of ovarian cancer. Beautiful landscape papers were published by first Alice Lee and Peter Park. Alice is here at the meeting, demonstrating again and again that this is something that is not a rare thing affecting one individual, but is in fact, again, common in cancers. At the same time, essentially, as these reports were coming out, our laboratory at Hopkins developed a robust monoclonal antibody for detecting the line 1 encoded protein. I think cancer researchers hadn't focused on the differential expression of line in a lot of malignancies because it wasn't incorporated in gene expression profiles, and any attempt to look at line RNA is complicated because of the great abundance of read-through transcripts. As we dug in and started looking for ORF1 expression by immunostaining tissue microarrays in collaboration with many of the faculty in the Hopkins pathology department, we saw this pattern again and again, where there was little expression in adjacent normal tissues, and then very high expression that you can see by this brown staining in a variety of these cancer types. So here are nests of colon cancer cells. Here's examples of lung cancer and pancreatic cancers. In pancreatic cancers, we walked backwards as well to look at early lesions, the so-called Pannin lesions that antedate the full-blown development of pancreatic ductal adenocarcinoma. We found induction of the protein in those lesions, and we see it along with collaborators, Yiming Shi's laboratory, very pronounced, induced in the so-called stick precursors that occur in the fallopian tube that are believed to be precursors of high-grade serous ovarian cancer. That's one of the most beautiful examples we have of ORF1 as a very binary marker. It occurs in these lesions exactly coincident with the development and fixation of p53 mutations. These observations inspired a graduate student in my lab to go on, Dan Ardelian did this work, and ask questions about what line expression is really doing to cell biology in malignant contexts or even in normal cell contexts. And this began to create for us an understanding of why line might be occurring, line de-repression might be occurring in these p53 compromised tumors. So one of the first experiments that Dan did was induce line expression in p53 competent cells, and what he saw in RPE cells, retinal pigment epithelial cells, was that the induction of line creates a very sharp decrease in clonogenic potential of the cells and a transcriptional induction of the p53 pathway. When he went to do cell fitness screens asking what gene knockouts allow for the continued growth and survival of line 1 expressing cells as compared to GFP controls, loss of p53 far outranked any other gene. So it really seemed that line expression in cells was prompting a p53 mediated response that slowed cell growth, arrested cell clonogenic potential, and that essentially for cells to continue to grow with line, they had to lose p53. It created an explanation for how line expression may create a gateway for p53 loss and the development of malignancies. On the flip side of that chicken and an egg problem, there's work by John Abrams' lab and others that p53 may serve as a direct suppressor of line transcription in many of the same cell types. Okay, so what happens if you compromise p53 and express line? And then do the same type of experiment. Look for genes that become important for the growth and development of these cells. So Dan went on to do exactly those types of screens. Here he took his RPE cells, had a very effective knockdown of p53, induced luciferase in a control arm, line in another, did a genome-wide knockout pool. And what he discovered was that very few genes advantage uniquely growth of line 1 expressing cells. One interesting example is APC. Loss of APC is especially good if you're a line expressing cell. What he recognized was that many new genes became essential for the growth of these cells, chief among them DNA repair genes, including many members of the Fanconi anemia complex that I'm showing here, as well as BRCA1. So these gene knockouts are relatively well tolerated in the control cells. These graphs show you abundance of the guide RNA over the 27-day course of the experiment. And the red line shows you the line expressing cells, which absolutely cannot tolerate the loss of the same genes. It suggests that when cells are growing, even if they've lost p53, they can't handle the sort of damage that's created to their genomes by unrestrained line 1 without these DNA repair pathways intact. We have exciting new studies that line is doing much more in tumors potentially than creating these beautiful surgical insertions of line, which frankly rarely seem to compromise directly tumor suppressor genes like our APC example. Rather, we have evidence that line is inciting truly broad-scale genome instability, at least at the levels that we are expressing in these experimental systems. So we can see with whole genome sequencing, just shallow, low-pass sequencing I'm showing you on this slide, an increased number of terminal deletions of chromosomes, amplifications, these undulating patterns consistent with chromothriptic events, subclonal losses of segments of DNA as we grow out clones from single cells, consistent with breakage fusion bridge cycles. It really appears that line expression may be a significant contributor to the abnormal karyotypes that are so broadly seen in all of these p53 mutated cancers. So again, I'll create the dichotomy of the rare case and what in fact is common in human disease. So it's clear from this early report of APC insertions that somatically acquired mobile element insertions can clearly mutate tumor suppressor genes and be overt cancer drivers, but perhaps this is just the rare patient. What actually is much more commonplace, what is, I think, a true hallmark of malignancies, is that there is an epigenetic dysregulation of line elements. We see this most pervasively in p53 deficient cancers, where its expression can perhaps be inciting in these cells essentially a mutator phenotype, and tantalizingly may impose new molecular dependencies or be an endogenous source of DNA damage that we might leverage in new ways of thinking about cancer therapeutics. So therapeutic implications for sure are important and exciting. We want to pursue these. But I'll tell you one more story about perhaps where we'll have our biggest effect on patients more soon, and that's with patient number 4. I'll describe her as our asymptomatic patient. She was a young 70 years old when she was diagnosed. She had an infectious laugh and shining eyes, lived near her childhood home with many of her extended family members. She loved her job as a teacher, but had recently retired. She'd been throughout her life an occasional smoker. She visited doctors several times in the months before she was diagnosed. Nonspecific symptoms: tired, bloated, abdominal pain that was poorly localized, a sensation one day that would go away the next, and somehow she always felt a little bit better on the days that she actually had an appointment to describe it to someone. The diagnosis came only after her disease was widespread throughout. The omentum had seeds of ovarian cancer. She represents one of 21,000 women who will be diagnosed with the disease this year. About 13,000 will die of the disease this year. Her blood went to a biobank at Penn before she started treatment, and then later was shipped to Boston for some studies that we are now conducting in collaboration with David Walt's laboratory to look for the ORF1 protein in the peripheral circulation. This is being spearheaded by Marty Taylor in my laboratory now at the Farber. So line ORF1p can be seen as this abundant marker in tissue samples, but by conventional ELISAs, we've been unable to detect the protein in the peripheral blood. It's now clear, though, that we can break that barrier with very ultrasensitive methods that enable single molecule detection. So here's an example of the type of assay that we're doing, Simoa assays for single molecule assays. There's a bead conjugated to an affinity reagent, a nanobody, or an antibody that binds to the ORF1p protein in a blood mixture that's added to this, and then a secondary antibody with a detection reagent. The fact that it's conjugated to a bead is critical because later these small beads are segregated into tiny wells, tiny areas of volume that allow for then the very high local accumulation of substrate from the ensuing enzymatic reactions. That gives you much more sensitivity to detect a single molecule as compared to standard ELISAs. This is our first kind of generation of this assay, and you can see in a cohort of more than 400 healthy individuals, we really don't detect ORF1p protein. We have instances in some cases where we know a contaminating individual who had a cancer was part of our healthy cohort. We have some others here that we are not able to explain. There may be natural variation. But you can see across different cancer types, we're able to detect ORF1 in a variety of these different tumors. The pie graphs show you the proportions. And here in our first group of 145 ovarian cancer samples, that's a particular tumor type where we're able to do relatively better than others. We're working hard on second-generation assays now for this protein with better affinity reagents that should lower our lower limit of detection. And you can see a few different versions of these assays here that show you varying degrees of characteristics comparing healthy controls with patients with advanced ovarian cancers like our patient. So at this point, I'll conclude this part of the story by just saying that line ORF1p may be something that proves useful as a marker in cancer screening endeavors, perhaps in high-risk populations, or perhaps in the general public, in patients for disease monitoring or for further patient stratification. We're working hard with different clinical teams to investigate the utility. So at this point, I'll conclude, and I hope that these vignettes have brought you up to date in what we're up to and what we're thinking about in human transposable element biology. I think it's an incredibly exciting time to be an investigator, to be a physician scientist in this field. I hope that while you're at Cold Spring for this meeting, that you come and take the time and have the patience, as Dr. McClintock asked us to, bring the openness to let new ideas come to you as we talk to one another. And that when you think about studies in human conditions for human patients, that this talk reminds you of the type of empathy that is brought to these studies, not an empty sort of relationship with patients that just stops there, but a type of empathy and urgency that engenders inquisitiveness to, as she put it, understand when something goes wrong with it, as we all try to develop a better feeling for the organism, a feeling for the human. With that, I want to thank members of my research lab and a tremendous group of collaborators who've helped us all along the way. Here on the left is a list of people who worked on these projects at Johns Hopkins very closely with me. I've mentioned a few of them as I went along. Dan Ardelian did all the self-fitness screen studies I described. Lisa Wong broke the problem of being able to map retroelements comprehensively in the genome. Lindsay Payer did all of our GWAS follow-up studies, a tremendously talented postdoctoral fellow. Nemanja and Marty, both pathologists in training at the time, did our ORF1p studies, and Marty indeed continues them in Boston today. A new cohort of people have joined my group since we transposed to the Dana-Farber. Many of them are here at this meeting and will be excited to be learning more about the field and hearing from you all, and a list of our collaborators here, past and present. So with that, I'll stop. Thank you.
Thank you so much. That was amazing. We definitely have some time for questions. Maybe I'll. Do you think line 1 might actually target some of these genes like P53 and APC that might help it transpose preferentially?
I guess so. I guess the question is, what do you mean by target? I think they are probably unlikely to be frequently directly mutating these genes. But it's possible both that these genes, P53 in particular, are direct suppressors of line. Also, I think our work in specific highlights that you need to lose vital tumor suppressors to continue to grow if you are a line 1 expressing cell. So all of these malignancies are late in life occurrences. They're not our pediatric ALL case. They're diseases that have long latency periods. And I think it's possible that an epigenetic dysregulation of line sets a stage for, indeed selects strongly for, the loss of these tumor suppressors, and that can create, the early hits in these malignancies. I think the fact that, for example, we see line integration sometimes mutating APC is very clear evidence that, at least in that case, the line activity is present before the loss of a critical tumor suppressor.
Kathy, absolutely beautiful talk. Can you now, with the sort of experience you've got, can you comment on the frequency or the propensity of line 1 to become activated in different lineages in the body? Is it equal in all lineages, or is it very biased, and why? And yeah.
This is a great and profoundly important question. I think we have tools to begin to address it as a field. I do have the impression that it is dependent on cell type, that we have a degree of activity, and I believe this is being borne out, at least in preliminary bioarchive studies, that we have a degree of activity in cells like colonic epithelial cells that's probably higher than what are now like published reports in neurons and other types of tissues. So I do think cell type matters. For the most part, we have not, as a field, although we absolutely have the tools to do it, identified the specific source elements that are active in these different cell types. I think long-read sequencing opens a tremendous opportunity to comprehensively map inherited line 1 insertions and identify those with hypomethylated promoters in different tissues, and to understand the inherent genetic variability that may exist in different individuals and in different tissues. I think we also now have very good tools as a field for doing single cell profiling of line integration sites, which will be crucial to address the question. In the past, most of our studies have relied on tumors to clonally expand a cell population. There's all sorts of abnormal things about that. But now we're, I think, able to take single cells and develop line insertion site maps and really directly measure a rate of retrotransposition and ask the question in different tissue types and across different individuals. It may also be very relevant to diseases outside of cancer as our eyes are open to this, but I think we have a lot of learning still to do. It's a great question.
Kathy, really beautiful story. I love that single molecule detection system and the results with ovarian cases, and it just made me wonder, it looked like you could detect it in 75% of the cases, and whether you know what was happening in the other 25%. Were they expressing L1 or were they not?
This is a great question. We have, with the first generation of the assay, been able to detect about 75% of women with very advanced, heavy burden of disease ovarian cancer. We know that when we take tissue blocks from women with the same types of tumors, but not the same cohort of women specifically, that we're able to stain for ORF1p protein and detect expression in about 90% of cases. It's possible that our second generation assay will now begin to sort of close that gap. It won't be a perfect test. We won't get 100%, we don't believe, because at least in 10% of women there's no expression of line. We don't know why that is. There are some cancer types, a great example is head and neck, where there's a group of head and neck malignancies that are driven by HPV and have relatively little line expression, another group that is P53 mutated that has relatively a lot, but we don't know the biology that makes that 10% of ovarian cancer cases different.
Thanks very much.
Hi, Casey. Here. Hansen here from Princess Margaret, Toronto. So I have two questions. So the first question is that it looks like the line detection can be a potential biomarker for diagnosis, and it looks like what you have introduced is mostly in the tumor detection. Can you comment on the liquid biopsy detection? And then the second question is that on top of the biomarker potential, is there a possibility for therapeutic targeting? For example, for the AR1B ALU insertion, would a CRISPR-based deletion or other method for the populations that have this genotype? Thanks.
Yeah, these are great questions. So the first is about liquid biopsies. Usually when people are describing a liquid biopsy for cancer diagnosis, they're talking about a large volume blood draw that then is frequently subjected to some sequencing-based analysis, either for detecting driving mutations in malignancies, in some cases detecting epigenetic signatures of malignancies. By and large, those are fairly expensive tests to run on samples and require pretty large volumes of blood draws. Our ORF1p detection method, in contrast, is a protein biomarker. And in all of the assays that I showed you, our input sample is plasma or serum, so it is a blood sample, but it's much smaller in volume than the liquid biopsy samples. It's like 25 microliters that's going into one of these reactions. We may be able to increase our sensitivity by just taking larger volumes of blood, in short, yes, it could be seen in liquid biopsies, but in fact is a much more straightforward, less expensive assay that requires much less blood. That may be part of its utility for following individuals over time or for screening en masse if we can demonstrate its utility. The second question is about therapeutic opportunities, and you were specifically interested then in inherited variants. I would say when it comes to an insertion at, for example, like the ARID5B integration, that's very hard to imagine as something that may be therapeutic, a good therapeutic target, because it is an incredibly common variant in human populations. The leukemias are still quite rare, and there's no evidence that I'm aware of that, once these diseases get going, that altering sort of the predisposing allele would make any difference. There are, however, some very promising studies. You may hear from Alice Lee and her colleagues at Boston Children's Hospital that in individuals with very severe phenotypes caused by inherited transposable element insertions, or rather germline insertions, there are different types of methods to try to restore the function of genes that they affect, and that there may be utility there. So I think that particular example is probably not a good therapeutic target, but the idea that we will be able to manipulate either germline DNA or mitigate the effects of germline variants is very real. So to be continued.
Two last questions. Short, please. First one then.
That was an excellent talk, Kathy. I was just wondering about the line 1 ORF1 protein expression again. Do you think that this is kind of the cell's strategy to kind of flag up cancerous cells to the immune system to be destructed? Do you kind of think that this is just a sign of chromatin dysregulation, expression of ORF1 protein, and the prevalence of this expression in ovarian cancer and perhaps other cancers could actually be beneficial to the host? Is there any evidence that these cancers are then targeted by the immune system because they're expressing line 1 ORF1 protein as a sort of novel antigen?
Yeah, this is a really fascinating question. There's a lot of interest in tumor antigens, as you're aware. As yet, I haven't seen very strong evidence that the line-encoded proteins themselves are being targeted by the immune system, although that certainly is a possibility. There's an emerging literature that is probably stronger for endogenous retroviruses serving in that type of capacity, and their ORFs and degenerating ORFs. Line expression in our experimental systems, we think probably through DNA damage, but we haven't tested it, can absolutely incite an interferon response. Your work in Hush and others have suggested that epigenetic dysregulation of the genome through a variety of mechanisms may create that type of signaling. So yes, I think it's absolutely possible that early in tumor development, this could be an anti-cancer mechanism, but exactly what's operative in what cell type, I think we have, again, a lot to learn about it. Thanks.
Last one. Is there one over there, or did it go? Okay.
Hello. My question is more towards like the tug of war of transposable elements and tumor suppressor genes, and I was wondering if there's like any example where there's a T that has been domesticated that actually helps the tumor suppressor genes in the protein level, or maybe like regulatory wise.
So there are many examples, I'm sure you're aware of some to ask the question, of transposable elements that are domesticated. Certainly a body of literature has suggested that tumor suppressor genes, probably the most work has been done on P53, have evolved, in part to silence retroelements, but I can't myself think of an example of a domesticated TE that specifically has that type of function. Cedric and others may have other ideas or things to propose in that category. I'm not sure.
Okay, great. Thanks to Kathy again.